Okay guys, in this video we're going to talk about the 5 different types of blood vessels, and then we'll talk about tissue fluid formation. And if we start with the 5 blood vessels then, so we're going to start with the arteries. So arteries have the thickest wall. So there's several different layers to the artery wall. There's the tunica externa, made of collagen. There's the tunica media, made of smooth muscle and elastic fibers, and the tunica intima on the inside, which part of that is the endothelium, which is very smooth and reduces the friction between the blood and the inside of the artery, the endothelium. So some arteries are called elastic arteries, like the aorta, which carries blood away from the left ventricle under high pressure. Now we know the elastic fibers in the smooth muscle, the tunica media, can stretch when the left ventricle in particular contracts. So blood's under very high pressure in that artery, and then they recoil once that blood is pulsed through that part of the artery back into their original shape. So that's the elastic fibers. Now the second type of blood vessel are the arterioles. Now arterioles are smaller than arteries and they branch off from an artery. So arterioles have a bigger cross-sectional surface area collectively versus a single artery. So that's going to allow more resistance to the flow of blood, and it's going to create a bit more friction because there's a bigger cross-sectional surface area, and therefore that's going to drop the blood pressure as it passes from the arteriole into the capillary. So the third one I'm actually going to do is the blood capillary. Now, blood capillaries are the smallest of the blood vessels, and the wall of the blood capillary is just made of endothelium. So it's 1 cell thick, and it's endothelium, which is a specialized epithelium. And the capillaries even further drop the blood pressure from the arteriole. Now we're going to get tissue fluid formation from the capillary, as some of the fluid from the blood plasma seeps across the fenestrations out of the capillary wall, and that tissue fluid is going to bathe the cells that make up the tissue. So we could put here the capillary, which is the smallest of the blood vessels. The wall is made of endothelium, and that's it. There's no smooth muscle, there's no elastic fibers. This is where tissue fluid is formed. Now capillaries then rejoin together, and when they do this they form a venule. So the venule will start to carry deoxygenated blood away from the capillary network and therefore away from the tissue. Venules have an even lower blood pressure than the capillary network because the lumen of the venule is obviously a lot bigger than the lumen of the capillary. And don't forget that the venule is furthest away actually from the ventricles of the heart, particularly the left ventricle. So the blood's actually had to travel a further distance as it's gone through the artery, arteriole, capillary, and then into the venule. So they've got a bigger lumen, and the blood has traveled the furthest distance away from the heart. So there's a further decrease in blood pressure. Now the venules then rejoin together to form veins. And we know veins have the largest diameter lumen, and they've got a smaller tunica media compared to the artery. They've got thinner walls compared to the arteries, and especially the veins below the heart, they can actually be squeezed by the action of skeletal muscle. And as the skeletal muscle contracts, it's going to push in on the thinner wall of the vein, and it's going to force the blood upwards through valves. So veins, especially the veins below the heart, do have valves, which allows unidirectional flow of the blood under very low pressure back to eventually via the vena cava back into the right atrium. So veins have the lowest blood pressure because the blood has travelled the furthest distance from the heart. They've got the biggest lumen size, and therefore there's least resistance to blood flow. So let's just put veins can have valves, especially the veins such as, and I'll do the biggest vein beneath the heart, which is the inferior vena cava. So that's going to deliver deoxygenated blood back into the right atrium. So let's just draw an artery and we'll compare that to the vein that we've got at the bottom, number 5. So an artery then is fairly regular in cross-sectional shape. I'll just put TS for transverse section or cross-section. And I'm going to do the inside endothelium. Now, whenever you do a scientific drawing, please make sure it's in pencil, not like me in pen. Use a sharp pencil. It's got to be a smooth line. There's got to be no shading and no sketching. Now, the very inside layer of the 3 layers is called the tunica intima. So when you label this up, okay, no arrowheads needed, but you're going to call it the tunica intima. Let's put that one there. Okay. Now, the thickest of the 3 layers is called the tunica media. So the layer in the middle here is the tunica media. Now within this layer you would find smooth muscle and the elastic fibres. Now, as we said a moment ago, what the elastic fibres do is during ventricular systole, when blood is being forced out of the left ventricle in particular through the aorta, as the blood passes this point in the artery, the elastic fibres are going to stretch, and as the blood's past the point now, they then recoil back into their original length, and that's going to help to maintain a high blood pressure. So we could put here elastic fibres. So the artery wall has a bigger, thicker tunica media, and this is made from smooth muscle, and don't forget the elastic fibres. So there's a thicker smooth muscle, tunica media, and there's more elastic fibres compared to the vein. Now the outside layer is called the tunica externa, so I could maybe do it this side here. So let's put tunica externa, and that's made from largely collagen. So collagen is a fibrous protein, and that's really important because it prevents overstretching of the artery as the blood pulses through that position. So remember, the thicker wall of the artery does a couple of jobs. It helps to withstand the higher blood pressure. The thick smooth muscle helps to maintain the higher blood pressure because of the elastic fibres, which we have at the bottom here. And some arteries, we get the smooth muscle can vasoconstrict and vasodilate. So if there's more blood going to pass through this point, the artery, the smooth muscle can vasodilate. And if there's a reduction in blood flow through the lumen, that's because the smooth muscle can vasoconstrict. Now, the very inside lining of the tunica intima, so this line here, is actually called endothelium. Now, endothelium is, sorry if I just put that down. There you go. Endothelium is a smooth surface which reduces friction between the blood and the inside of the artery, the endothelium. Now, the capillaries are only made of endothelium, whereas the artery is made from the tunica externa, the tunica media, the tunica intima, of which the very inside layer is the endothelium. Now, if we have a look at a vein has the same sort of layers. So it's got the tunica externa, tunica media, and tunica intima. However, it's got not quite the same regular appearance if we do a cross section. So I'm going to draw my vein a bit like this. So it hasn't got a regular appearance. So this is the vein in transverse section, cross section. Now, the lumen is obviously going to be a bit bigger. So when you draw a vein, what you should do is the thickness of the layer should be consistent all the way around. So you shouldn't have a thicker bit one part and a thinner bit in another part. This is the lumen. Now, the tunica externa is again made of collagen, which is a fibrous protein. So please, when you draw this, try and get the layer, same consistency all the way around. It does have a tunica media. So this is going to be the bit here made of smooth muscle. However, it's a lot thinner than the artery. So we can now label this one up. So this would be the tunica externa, made of collagen. Obviously, just do write this out in the exam. I'll just abbreviate it for today. This is the tunica media. So it does have some smooth muscle, but it's a lot thinner than the artery. And again, it's got the tunica intima on the inside. And the very inside layer of the tunica intima, as we said earlier, is the endothelium. So there's a much reduced blood pressure inside the vein versus the artery. Now, what happens is if these are veins beneath the heart, skeletal muscles can contract. For example, in the calves, these skeletal muscles will surround the veins in the lower leg. And as the skeletal muscle contracts, it's going to squeeze in the vein, because obviously there's a thinner wall, so it can be squeezed in much more easily than an artery could do. So there's a massaging effect of the skeletal muscle. So we could say here, skeletal muscle contracts. And what that does, it actually forces the blood up inside the lumen of the vein. So I'll just go up here and then we'll have a look at this time. Imagine you had a vein from the side here. So I'll just do it like this. So this is our vein looking from the front this time. And there are valves. So I'll just do the valve here. And as the blood gets squeezed upwards, the valves are going to open like this. So the blood's going to go up and through the valve. So veins can have valves. But what happens is the blood falls back into the pockets of the valve, and that snaps shut the valve. So there's no backflow of blood back down the vein. So essentially what happens is the blood moves up in stages through the veins back up to the major veins like the inferior vena cava, and therefore back into the right atrium eventually under, remember, a very low blood pressure. Okay. Now, we need to talk briefly about tissue fluid formation, and that happens at the capillary. So what I'll do up here, I'll do a little capillary. So this is my capillary. So I'll just put here, capillary. And one end of the capillary, so I'll do the end on the left here, it's called the arterial end of the capillary. Arterial. And the other end of the capillary is called the venual end. Make that a bit bigger. Now remember, the wall of the capillary is called endothelium. So there's no tunica externa, or there's no tunica media, or no tunica intima, it's just endothelium. Now with the arterial end of the capillary, there's a higher hydrostatic pressure versus the osmotic pressure in the blood plasma. So the hydrostatic pressure is due to the blood coming from the left ventricle, from an artery, and then the arteriole into the start of the capillary. So the hydrostatic pressure is a pushing force of the fluid, the blood plasma, against the wall of the capillary. Now, because the blood plasma does have dissolved solutes like glucose and amino acids and ions, it does also have a negative water potential. So the negative water potential actually wants to draw in a fluid or water by osmosis, because that's going to be down a water potential gradient to a more negative water potential. But it's just that the hydrostatic pressure, the pushing force of the fluid against the inside of the capillary, is greater than the pulling force of the osmotic pressure at the arterial end of the capillary. So the higher hydrostatic pressure is having a bigger impact, a bigger net impact than the osmotic pressure. So the net effect is to force out water and any dissolved solutes within the water, such as glucose, amino acids and ions, through the fenestrations, which are little gaps and holes in the endothelium. So we're going to get, and I can just put this down here, look, fenestrations in the endothelium of the capillary wall. They're little gaps between or through actually the endothelial cells, and the high hydrostatic pressure forces out the water and dissolved solutes through the fenestrations, and that then becomes tissue fluid, which is going to bathe the cells that make up the tissue. Now remember, the definition of a tissue is an aggregation or collection of similar specialized cells that all work together to perform a certain function. Now, if we have a look at the capillary, as the blood passes down from left to right through the capillary towards the venule end of the capillary, because of the loss of fluid from the blood plasma, there's going to be a drop in the hydrostatic pressure. So we could put here, the hydrostatic pressure inside the blood capillary goes down because there's less fluid inside of it, because that's become the tissue fluid. However, we have to remember that inside here, there are bigger plasma proteins that could not get through the fenestrations of the endothelium. So these plasma proteins remain in the blood plasma because they're too big to fit through the fenestrations. Now, because there's less fluid, there's relatively a higher concentration of plasma proteins at the venule end of the capillary, because there's the same number of plasma proteins because they could not escape through the capillary wall. But the smaller volume of fluid means their relative concentration is actually higher. So at the venule end, there's actually a more negative water potential value in the blood plasma compared to at the arterial end. So because there's a loss of fluid as tissue fluid, the hydrostatic pressure drops at the venule end. There's the plasma proteins that remain in the blood plasma, therefore there's a more negative water potential. So at the venule end here, we could say this time that the osmotic pressure, which is a pulling force wanting to draw water in to the blood plasma, has a bigger effect than the hydrostatic pressure, which is dropped. So at the venule end, what happens is water, which is from the tissue fluid, gets drawn into the venule end of the blood capillary by osmosis down a water potential gradient back into the venule end of the capillary. So about 90% of tissue fluid gets drawn back into the venule end of the blood capillary by osmosis down a water potential gradient. And please remember, obviously within that water there's dissolved carbon dioxide, there's metabolic waste such as urea, for example, if we're talking about the liver. Now, the other 10% of the tissue fluid, rather than drain back in by osmosis into the venule end of the blood capillary down a water potential gradient, the other 10% drains into a lymphatic capillary. So a lymphatic capillary will pick up around about 10% of tissue fluid, and that's really important because imagine there was an infection, for example, by a bacterium or a virus within that tissue. The tissue fluid is going to take some of the bacteria or some of the viruses, and it's going to drain through the lymphatic system, and that's going to go through the lymph vessels. And along the lymph vessels are lymph nodes, and in the lymph nodes you find B cells and T cells, and they're going to have an immune reaction against any potential antigens that might be present. That's why sometimes when you get an infection, your lymph nodes swell up, because you're getting immune reactions against the antigens, the bacteria and the viruses within that lymph fluid. So it's important to drain our lymph fluid to make sure that it goes through the lymph nodes, and therefore there might be an immune reaction against any potential pathogens that we want to destroy via the cell-mediated or the humoral response. Now, the humoral response will involve production of antibodies from plasma cells against, obviously, the specific antigen.